Good afternoon, everyone. My name is Sebastian Fica. I study spliceosomes at the University of Oxford. And today we're going to have an interesting webinar on the role of spliceosomal small nuclear RNAs in splicing and how this goes wrong in rare disorders. And the seminar will be delivered by Dr. Nicky Whiffin from the Big Data Institute, also at the University of Oxford. So over to you, Nicky.
Perfect. Great. Well, it's great to be here. I'm going to talk a little bit today about our work on spliceosomal small nuclear RNAs and their role in rare genetic disorders. So just to firstly orientate those of you who don't work in this field. So in the UK, we define a rare disease as one that individually affects fewer than 1 in every 2,000 individuals. But there are thousands such rare disorders identified. So collectively, they're actually very common, affecting 1 in 17 individuals or over 300 million people worldwide. They're often very severe and life-limiting, and over 80% of them are thought to have a genetic basis. And actually identifying that genetic basis of the precise DNA modification that leads to disease is incredibly important to that patient and their family, both for clinical management, for access to research, clinical trials. Sometimes it's the first diagnosis a patient actually gets. So it tells them information about why they have a particular disorder or difficulties. And it also allows family planning. So allows us to screen additional family members or give reproductive advice on recurrence risk. And finally, it allows them to access a supportive community of people going through similar challenges. But despite the huge value of getting a genetic diagnosis, the majority of patients with rare disorders currently may remain undiagnosed. So this is data from the Deciphering Developmental Disorders Project, or DDD, that's been running for about 10 years. And they recruited a set of thousands of individuals with severe developmental disorders in the UK. And in their project, they have a diagnostic rate of about 40%. So around 60% still don't have an answer. But our current approach to genetic testing, both in clinical settings, but also the approach taken by DDD, is very focused on regions of the genome that directly encode proteins. So we know that our genome has about 3 billion base pairs of DNA, but only a subset of that directly encodes proteins, so about 1.5% of it. And that is where our clinical genetic testing focuses. But today I'm going to focus on other elements of the genome, so namely non-coding RNAs. So there are at least 25,000 of these annotated across the human genome. And when we're talking about rare disease diagnoses, these genes have been pretty much unstudied. And one of the key reasons for that is that we have historically just created data that uses either panel or exome sequencing, and where we only directly sequence the regions of the genome that directly encode proteins. But that has been shifting. So in the UK now, we use whole genome sequencing in routine clinical care. And this was an initiative first started under David Cameron's government, where the UK decided to try and be a leader in genome sequencing and created a company called Genomics England that first hosted the 100,000 Genomes project that you might have heard of. And that data from that project and now the point of care genetic testing done within the NHS all goes in this thing called the National Genomics Research Library, which is a core resource that we use in our team's research. But crucially, this data, this large number of individuals that have had their entire genome sequenced, gives us a really important resource to be able to study variants in non-coding regions of the genome. So specifically, narrowing down further from all of the non-coding RNAs, of which there are many across the human genome with very diverse effects, today I'm going to talk specifically about the small nuclear RNAs, or snRNAs. And these genes are very critical components or catalytic components of the spliceosome. And so they mediate the sequence recognition piece in guiding the spliceosome to the correct splice sites in order to mediate the removal of the intron, the splicing together of the exons into the final mature RNA transcript. So my interest in these genes actually started when a student in the team, Yi-Yang Chen, identified actually a single specific variant. And I'm showing this here by a tower of triangles. Now, he wasn't specifically looking at these small nuclear RNAs. He was actually looking at potentially novel translated elements detected through ribosome profiling studies. And it just so happened that this particular variant was annotated as a potential stop loss of one of those potential novel translated ORFs. But nonetheless, it turns out that that potential ORF has nothing to do with the mechanism here. But we're very interested because he identified this very recurrent single base insertion in the middle of this gene called RNU42, which is only about 145 base pairs long. And I was initially very surprised at this. I didn't believe this could be a true result. I thought this was more likely to be a sequencing error. And that is because all of these variants were identified as what we call de novo. That is, they were newly arisen in a child and didn't exist in either of their parents. And these de novo variants are thought to be random variant occurrences, so we expect them to be relatively evenly distributed across the genome. So to identify around 40 individuals here with exactly the same change was somewhat surprising. But Yang was somewhat persistent, and he said, hang on a minute, all of these individuals have neurodevelopmental conditions that are all previously undiagnosed. And when we looked further into this result, we could see that this variant existed within a portion of this gene, which is depleted of variation in the general population. So on the bottom here, I'm showing you as these dots here are variants that are found in 500,000 individuals from the UK Biobank population cohort, where the length of the lollipop sticks are the frequency of the variant in that population. And you can see this region pointed to in the middle, where there are very few of these dots compared to the rest of the gene. And all of those that do exist are incredibly rare. They don't have any lollipop sticks. And this defined an 18 base pair region, where when we looked again back into our National Genomics Research Library or Genomics England dataset, we found additional patients that also had different de novo variants also within this 18 base pair region. We then reached out to various collaborators and used a tool called Matchmaker Exchange, and very quickly identified a cohort of 115 patients, all who had previously undiagnosed neurodevelopmental disorders with de novo variants in this incredibly small region of this non-coding RNA. And the thing that remains true across all of these cohorts and all of the cohorts that have been studied subsequently is that around 70 to 75% of all individuals still have that exact same single nucleotide insertion that Yi-Yang first identified. And we don't know what is causing the recurrence of this single variant. It remains a bit of a mystery. So we then moved on to characterize this disorder with some collaborators, particularly Anne O'Donnell-Laurier and her team at the Broad Institute in Boston. And we characterized the disorder caused by variants in this region as a severe neurodevelopmental syndrome. So individuals have global developmental delay and intellectual disability that is marked as either severe or profound in most cases. The condition is very life-limiting. These individuals are mostly non-verbal. They have difficulties feeding, brittle bones, slow muscle tone, and around 70% of them also have seizures. And working with the families, we then named this syndrome Renew syndrome, speaking to the renewed hope on getting to a genetic diagnosis. So why is this tiny RNA so important? Well, RNU42 encodes the U4 snRNA, and the specific role of U4 within splicing is to help with recognition of the 5' splice site. Oh, skip slide. So in the spliceosome, U4 is very tightly wound to another snRNA called U6, and it is U6 that directly binds to the 5' splice site. And the U4, U6 duplex has to be unwound in order for splicing to be activated. If we just zoom in on that interaction between U4 and U6, here I've got U4 in red and U6 in yellow. And this short sequence here of letters is the echo-gaga sequence, and that is what binds to the 5' splice site. And the variants that we find in individuals with Renew syndrome are in two structures, the quasi-pseudoknot T loop here, or the stem3 region up here, that are thought to correctly position that echo-gaga sequence of U6 to correctly receive the 5' splice site. And consistent with that known biology and the role of U4 in 5' splice site recognition, when we looked at RNA sequencing data also available within the NGRL for Renew patients, shown here in teal, compared to controls, we see an elevation in significant events that correspond to usage of alternative 5' splice sites. And what I mean by that is, instead of correct splicing at the correct canonical 5' splice site, in individuals with Renew syndrome, we see a splicing either a little bit downstream or a little bit upstream of that canonical site. And this change in 5' splice site usage is not random, it appears to be systematic. So here what I'm showing you is an incredibly boring histogram where the grey bar here is showing that when we take our control individuals, how often do we see the same different 5' splice site event as significant in two different individuals? And that's basically never. And if we compare that to our individuals with Renew syndrome, we would currently see the same events differentially spliced in these individuals. So we then went further to look at the splicing motifs surrounding these 5' splice sites that were differentially used in cases versus controls. And on the bottom here, what I'm showing you is this, the motif surrounding the 5' splice sites that are decreased in usage. And this is basically the canonical 5' splice motif. And on the top here, what I'm showing you instead is the motif around sites that are increased in usage. And just to draw your attention to these three bases here, the +3, the +4, and the +5 position, as these are the positions where we see a shift in the motif with an increase in T at 3 and 4 and an increase in C at position +5. And interestingly, these three positions of the 5' splice site are the exact three positions that directly bind to that U6 echo-gaga sequence in 5' splice site recognition. From follow-up work here by Caroline Naver and colleagues that was published last year, they showed that the actually extent of splicing disruption correlates with phenotypic severity. So what I'm showing you here is a principal components plot where they took a set of different controls, shown here in purple, and a set of individuals with Renew syndrome, shown in red and teal. And they looked at all the sites, the 5' splice sites, that were significantly different in usage across the entire control set and the entire case set. They took those significant splice sites and they plotted basically the proportion spliced in, so the usage of each of those different splice sites, and used that to create this PCA plot. And as expected, you can see that this separates out the cases versus the controls, but you can see that the individuals with a more severe phenotype, shown here in red, are further separated from the control individuals than these teal individuals with a milder phenotype. So that's really nice. We expect that these variants are causing disease through this disruption to a kind of global but low-level disruption to 5' splice site usage. But one of the things that's perhaps most surprising still to me, even a couple of years after we first identified Renew syndrome, is the frequency at which RNU42 is a cause of neurodevelopmental disorders. Now, it's incredibly hard to define the frequency of a disorder which needs genome sequencing to identify it, because historically we have not had individuals having genome sequencing as a first-tier diagnostic test. But we tried to estimate this, and we used that diagnostic rate from the Deciphering Developmental Disorders cohort of 40%, and assumed that those individuals would not get into our cohort, and use that to estimate that the frequency of individuals with NDD explained by RNU42 variants is about 0.4%. Now, if we compare that to the genes that are the most common diagnoses within the DDD cohort, we can see that it's not much lower than these other genes, which have frequencies of around 0.55 to 0.7%. But the really key difference here is that while these three other genes are enormous protein coding genes, in excess of 9,000 base pairs long, RNU42 is a tiny non-coding RNA of only 145 base pairs. And not only that, but all the variants we identify are only within 18 base pairs of DNA. So there's a very different amount of hit space for those random de novo variants to occur. And because these other genes are very large protein coding genes, they were first identified when we first had access to large exome sequencing cohorts that profiled protein coding regions of the genome back in the early 2010s. But in contrast, given that RNU42 is a non-coding RNA and requires genome sequencing, it took us to 2024 to identify it. And we now estimate there are about 100,000 individuals worldwide who have Renew syndrome. So this has been an incredible story. I like to joke that I have found the secret to getting your research published in a national newspaper, and that is all you need to do is diagnose the son of one of the editors. But jokes aside, this is a beautiful article that was written by James Coney about his son Charlie and his family's journey to a diagnosis. So if you haven't seen this article, then please do go and check it out. It's a really lovely article. But the two things that I pulled out at the bottom here, two quotes, are things that we repeatedly hear from families about the difference that a genetic diagnosis makes to them. Firstly, from Charlie's mum, Sarah, it's nice to know it's not our fault. So a lot of parents with children with severe developmental disorders think that it was something they did, maybe during pregnancy, that led to their child's disorder. And a genetic diagnosis is the first time they're told that it was just chance. But also suddenly these parents go from raising their child in the dark without a handbook, not knowing what the future looks like, to suddenly being part of a community. There are now over 700 family members on a Facebook group where these families can share their stories and share advice and leading to them feeling no longer alone. So that's really powerful. So there have now been hundreds, if not thousands, of Renew syndrome diagnoses around the world. Here's a map that has been created by Renew Syndrome United, which is a family advocacy foundation founded by some mums in the US. And here, every dot on this map is a diagnosis of Renew syndrome. So this map simultaneously fills me with joy and horror. Joy at how many dots are now on this map, but horror because it very acutely tells us about the disparities in genetic diagnosis. And here, where the dots occur on this map are where we have better access to genetic testing and particularly genome sequencing. So we have a lot of work to do to try and get diagnoses of Renew syndrome to people all around the world. But nonetheless, there are now family foundations, not just in the US, but that have been founded in various different places all around the world, and even more are coming online every month or so. And back in last year, so pretty much exactly this time last year, I had the pleasure of going to New York, to Long Island, to the very first Renew Hope conference organized by Renew Syndrome United, which is spearheaded by this woman on the right here called Jessica, whose son Devon, shown in the middle at the bottom, has a diagnosis of Renew syndrome. And they managed to get over 200 people in one room, families, kids with Renew syndrome, researchers, and also pharma companies, all who have an interest in Renew syndrome. And it's a pretty amazing event. So that was a bit about our initial discovery and how we got into this space. But there were still quite a lot of unanswered questions at that point about the kind of variant landscape across the entire RNU42 gene. So we worked with two incredible experimentalists, Greg Findlay, who's a group leader at the Francis Crick Institute in London, and Joachim, who was a postdoc in his lab at the time. And here we tried to map the variant effects across RNU42 using a saturation genome editing approach. And so what do I mean by that? Well, here, Greg and Joe made a series of different edits using CRISPR-Cas9 genome editing into a set of cells, such that each cell has a different variant, And then they watch those cells over a period of 10 days to see how many of them survive, sequence those cells in order to look at the effect of each individual variant on cell survival. And we use these function scores to measure that effect. So because RNUopathy is such a small gene, as I said, only 145 base pairs long, we could assay every variant, or variants across the entire length of the transcript in one experiment. So we did every single possible single nucleotide variant across the entire transcript. Focusing on this 18 base pair region in the middle that we know is important for Renew syndrome, we also did every single possible single nucleotide insertion and deletion. And then conscious that we needed to be able to separate out variants on an axis of deleteriousness, we also spiked in some variants we thought might be particularly deleterious, so multi-nucleotide insertions at the positions where we see the majority of variants in Renew syndrome, and also some control insertions that we didn't think would be deleterious into these loop regions shown here in yellow. And this is a plot of what the data looks like. So here we've got the nucleotide position across the gene shown on the x-axis, and each dot here is a different variant, and the shape of the point is the type of variant, with the majority of these being circles because they're these single nucleotide variants. And the color of the point here is corresponds to where we see the variant. So the ones in red have been observed in individuals with Renew syndrome. The ones in blue have been observed in individuals in the general population, so in either the UK Biobank or the All of Us cohorts. And the ones that are in teal, we've never observed in a human that we've sampled so far. And on the y-axis here, we have the function score, with the further down the plot the variant is, the more of an effect it had on cell survivability, so the more deleterious we infer the variant to be. And as you can clearly see, this shaded grey region in the middle is our 18 base pair Renew region, where you can see that the majority or a large proportion of variants are, including all of the variants we find in Renew syndrome, are dropping out or deleterious in this assay. So what did we learn? Well, one of the first things is that I just said, all of our Renew syndrome variants in red were below the line of significance, which is this dotted line, showing they were significantly depleted in this assay. So they had a significant effect on cell survivability. And that is in contrast to the majority of variants that are seen in the general population, shown here in blue. If we zoom in on the 18 base pair region that we know is important in Renew syndrome, the boundaries of which are shown by these solid grey vertical lines, we can see that actually we can refine this region to two distinct regions, one of 9 nucleotides corresponding to the T loop, and one of 4 nucleotides corresponding to the STEM3 region, with the variants in the middle actually not being deleterious. What we then did is we separated these variants into 2 different categories, the ones that we identify in Renew syndrome, so ones that were either that were strongly depleted in our assay using a arbitrary cutoff, and ones that were only moderately depleted in our assay that were between that and the line of significance. And what we could then do is see if we could see any differences between individuals carrying these different variants in terms of phenotypic severity. And this plot is a little bit difficult to see, so I'm going to focus on one simple axis here, which is the axis of speech. And here we can see that individuals that have a variant that is strongly depleted or strongly significant in our assay, over 90% of them are completely nonverbal or can say only a few words. And that's in stark contrast to individuals that have a moderately depleted variant, where the vast majority, so 95% of them, can speak simple sentences or have completely normal speech. So here, this is, to me, is quite remarkable. This is a simple assay of cell survivability actually done in a haploid cell line. I should have said that, hap1 cells, can actually separate out variants on an axis of phenotypic severity. We also can show that these SGE scores correlate with the extent of splicing disruption. So here is a very similar plot to what I showed you earlier, although for some reason this one is flipped around. And here, this is that PCA plot based on the 5' splice site, significant 5' splice site events, with the controls on purple on the right, and the cases coloured by their moderate and strong categories. We can see here the individuals with a strongly depleting variant are again further separated from the controls than the individuals with moderately depleting SGE variants. We also show that this saturation genome editing experiment massively outperforms in silico prediction tools that are designed to estimate the deleteriousness of variants. We don't have many such tools that work on non-coding regions and non-coding RNAs, but one that does is, or tries to, is CAD. But CAD has quite a heavy reliance on things like cross-species conservation, and the entire sequence of RNU42 is very highly conserved. So if you can see on this plot on the right, the CAD score here is on the x-axis, and there's pretty much no delineation in the CAD scores for these variants across the entire gene. And if you took a CAD score threshold that would capture all of the Renew syndrome variants, it would also classify over 70% of variants across the entire gene as likely deleterious. So it's useless for anything clinical variant classification. In contrast, our SGE score, which is shown here on the y-axis, can actually separate out variants that are deleterious from the majority which are likely to be benign. But the eagle-eyed among you might have noticed when I first brought up this slide that it's not all going on in the center of this gene. There are actually 4 other linear regions which correspond to 3 structural regions where we see variants also depleted in this assay. But in contrast to variants in the Renew region, where they're all either red or teal, so they're either observed in patients or they're never observed in anybody, most of the variants in these other regions are actually in blue. That means we find them in population controls. So what is going on here? Well, Greg very quickly hypothesized that maybe these variants were recessive. And indeed, all of these observations in controls were actually heterozygous variant calls. And when we looked into this further, this is work done by Rossio in Australia and Alex Blake, who at the time was in Manchester and is now with my team in Oxford. We identified a set of different of patients with homozygous and compound heterozygous variants in these 3 different structural regions. So stem 2 up here, the K10 and the 5' stem loop down here, and the SM protein binding site on the right here. So these 3 other structural regions that are distinct from these central regions shown here in teal, where we find variants in Renew syndrome. So Renew syndrome, dominant de novo variants. These other regions, we find these recessive, 1 inherited from each parent variants. And we see that this recessive NDD is distinct from Renew syndrome. I should say there's a lot of overlapping features. So they have a similar rate of seizures. They have a similarly severe global developmental delay and intellectual disability, but there are some key differences. And perhaps the most striking differences are in the brain MRI phenotype. So we see a significant enrichment of abnormality of the cerebellum, particularly cerebellar atrophy in individuals with the recessive syndrome. And also we see not significant enrichment, but abnormalities of the cerebral white matter. And in Renew syndrome, we have very heterogeneous changes in white matter that have been recorded in patients. In contrast, in this recessive NDD, there is a very specific white matter phenotype. I will caveat this here with the fact that I am in no way an MRI expert. This is the first time I've ever looked at MRIs during this project. But I'm told that these yellow arrows are pointing to a dilation of perivascular spaces in the periventricular and deep white matter, otherwise known as these grey dotty regions that you can see at the end of these arrows. So this is actually, I'm told, a very striking MRI phenotype, so striking that actually many years ago, Kaz Simmons, our collaborator in Australia, had clustered a few patient families based on this MRI phenotype, hypothesizing they would have the same underlying causal gene. And after we'd identified biallelic RNU42 variants in one of those families, he went and searched in a second and again found biallelic variants in RNU42. So showing just quite how striking that MRI phenotype is. So we think that the mechanisms underlying these dominant and recessive disorders are also distinct. So here what I'm showing you again is that PCA plot here, colored slightly differently. Here we've got controls in red. We've got Renew cases in teal, and then plotted onto this, we've got our recessive individuals in red. And as you can see, these recessive individuals are clustering with the controls, so that we don't see the same systematic change in 5' splice site usage that we see in individuals with Renew syndrome. rather, when we look at these individuals with a recessive NDD, we see a loss of function or a dramatic loss of RNU42 transcript levels. So here on the left, I'm showing you RNU42 on the y-axis and RNU41 on the x-axis. And you can see these red recessive individuals are lying pretty much on this x-axis. And this is plotted out as in a different way on the top on the right here, where we can see a significant reduction in RNU42 transcript levels in individuals with the biallelic variants. And we don't see this in Renew. And actually, correlated with this, we see a dramatic upregulation of RNU41 levels, the paralog gene to RNU42. This was interesting to me because I expected that these two paralogs could compensate from each other, but the fact that this upregulation is happening, but these individuals still have an NDD, is perhaps suggesting something more complex is going on here, which is very interesting. So we have this working model that these different disorders have different mechanisms underlying them, which is why they are distinct. But to add even more to the picture, there has been another paper that was published at the start of this year showing that variants in this central loop region, shown here in yellow, and also in the corresponding positions in the U6, cause retinitis pigmentosa, so a retinal phenotype. So here we have 3 distinct disorders caused by variants in distinct regions of the gene through distinct mechanisms, which I think is super fascinating, especially given all of this is going on in just 145 base pairs of DNA. But I should say that all of this is not limited to the story around RNU42. And also RNU42 was not the first of these snRNAs to be linked to a neurodevelopmental disorder. There have been 2 genes that had been linked prior. So back in 2011, the first link was made between RNU4 attack that functions in the minor spliceosome and rare recessive disorders. And a few years later, in 2017, RNU12, also in the minor spliceosome, was linked again to rare recessive disorders. But since our identification of Renew syndrome in 2024, there have been a bunch of subsequent discoveries. So RNU22 and RNU5B1 were both then linked to dominant disorders. And around the same time as we found the recessive disorder in RNU42, there was a very frequent recessive disorder associated with variants in RNU22. As I mentioned, RNU42 and different U6 paralogs have been linked to retinitis pigmentosa. And to add to that, RNU6 attack, again in the minor spliceosome, has been linked to recessive neurodevelopmental disorders and also autoimmune diabetes. So this has been an incredible pace of discovery with many new gene disorder links made just in the space of a couple of years. And these disorders we kind of collectively term now the RNUopathies. So these RNUopathies are a significant cause of neurodevelopmental disorders and explain a significant fraction of individuals with previously unsolved neurodevelopmental disorders. This is data from a subset of the NGRL, so the 100,000 Genomes Project, where these new disease links explain about 1.5% of previously undiagnosed individuals within this cohort. Now, the majority of this is driven, well, I say majority of this, just under half of this is driven by a Renew syndrome, but a significant fraction now from the recessive RNU22 disorder, with smaller fractions due to the dominant RNU2 disorder, RNU5B1 and RNU42 recessive. But I really think we're only just at the tip of the iceberg here, and there'll be many more discoveries in this space in the next few years. So just to finish off with a little bit of a look to the future, so we and many others are doing a lot of more research into these RNUopathies. We now have a lot of cell line models, both that have been that are patient-derived, but also that have been engineered using genome editing. We have mouse models for Renew syndrome that have been established with the help of Jackson Laboratory in Maine. And there is a lot of progress towards looking for treatments that might be able to treat Renew syndrome. So this is quite difficult. Most rare disorders, we really struggle to have any development of treatments, but Renew syndrome actually passes the frequency threshold, which is needed for pharmaceutical companies to become interested. And also, if you remember back to the very start of my talk, the predominance of this same specific genetic variant means that we can design therapies that target that specific variant, which makes it more attractive than trying to design a whole bunch of different therapies to target the different variants in more heterogeneous conditions. So the future is very bright. And I can't finish without flashing up again some pictures of the beautiful Renew syndrome individuals. And their families have gone from not knowing anything and being completely in the dark and not knowing what the future holds to actually having some renewed hope that perhaps at some point in the future there might be a treatment for Renew syndrome. And so with that, just to finish by thanking, this slide is nowhere near comprehensive. There have been a huge number of individuals involved in all of this work, but just to point out a few key individuals, so Yi-Yang, without whom we wouldn't have ever got into this space, Greg and Joe for their beautiful experimental work for saturation genome editing, and Alex, Kaz, and Rossio for their key roles in defining the recessive disorders, and always have to thank the patients, their families, and their clinical teams, without whom none of this would be possible. And happy to take any questions.
Right, okay, so we do have one question that somebody submitted, but I kind of want to start by maybe asking one myself, which is, there's many different variants for all the snRNAs, and my question, I guess, is, do you see any kind of mutation, like a lot of these mutations that occur in specific variants for other snRNAs that don't really lead to kind of to any of these types of syndromes, or because it seems that you have a very specific, if you, like in the case of RNAu4 versus 2, depending on which one you kind of affect, you might have, you have always kind of this neurodevelopmental syndrome, but then in the case of U1, of the RNAu4-1, even though it's expressed kind of mostly everywhere, right, you don't really get a very kind of localized sort of phenotype. And so I'm kind of wondering whether, there's one of them, like specific variants are maybe more prone to mutation than others because of where they're expressed.
Possibly, I think this is a complicated picture based on, one, do the variants occur in the first place, which I think might be kind of what you're saying.
Yeah.
Also, whether they're expressed enough for a phenotype to be caught.
Right.
So the primary variant we see in Renew syndrome does also occur in RNU4-1. There's 6 individuals in the UK Biobank that have that variant,
yeah,
but they don't have any phenotype in common. So I think that's because RNU4-2 is the predominant isoform in basically every tissue we've looked at so far.
I see. Okay, right. That was part of my question.
Mouse, which is kind of fun for doing some of our mouse work. But
so actually, as a follow-up maybe to that, do you think then, because almost all the snRNA genes have multiple actually copies in the genome, which to me is a bit, I don't know, weird compared to yeast, for example, where there's like one copy and you make the snRNAs and you're done. So do you think it's partly a backup mechanism? Because I guess your work on RNAu4-1-1 and -2 suggests partly that there is a bit of compensation, right? Because when you have mutations that disrupt like SM binding and the level goes up for one, the other one kind of seems to be upregulated.
Yeah, it looks like there's at least some compensation at the expression level. Whether that's functionally compensating or not is the thing that's currently unclear.
Right.
It's very interesting that in the kind of U6, the U6, there's, I don't know, this is all still very uncertain and changes every week, it seems, but there are potentially 5 U6s that are kind of equivalently expressed.
Right, yes, yes.
And in retinitis pigmentosa, you see exactly the same variants across 4 of those 5 genes that cause RP. So clearly a defect of, say, 20% of the U6 pool is enough to cause a retinitis pigmentosa phenotype. But in contrast, for neurodevelopmental disorders, we haven't yet identified any link to U6 because we think there will be enough compensation to kind of prevent that happening. Whether you could have multiple mutations across multiple different U6 copies and then get an NDD is something we haven't yet elucidated.
And so for U6, just a kind of to wrap up, and then I'll get to the attendee question. Do you think, so for U6, do you see actually the same kind of, say, skewness to expression? There's one variant that gets expressed mostly everywhere, and the other ones are like low levels, maybe peppered around, or is it more kind of a broader distribution compared to what you see for U4 snRNA, for example?
So I kind of want to caveat this with the fact that we haven't necessarily looked at this in the best way yet, because the majority of our sequencing cohorts, our transcriptomics cohorts, are poly-A selected. And as you know, these genes are not poly-A.
Yeah, exactly.
We have resources like GTEx, which are cross tissue, but we can't use them to look at snRNAs.
Okay.
So we're creating that data. We're trying to understand also all the single cell stuff that we'll be able to look at. Kind of, people always ask me which cell type in the brain is the one that's important. So we don't know, because all the single cell stuff is poly-A selected. So one day we might know the answer.
Do you think there's actually a way to look at the actually transcribed snRNAs and do the sequencing in reverse so that you can actually look at have people try to look at this? If it's a problem, because the poly-A seems an issue, right? So how are you gonna, I mean, you can do site selection and all this kind of stuff, but
yeah, I mean, we just need ribodepleted protocols, basically.
Yeah,
I don't think anybody's created a ribodepleted poly-A, a ribodepleted single cell protocol at this point. So
yeah, okay, fair enough. So we have only one question. Somebody was asking if the severity of disease affects life expectancy, or is it just a matter of quality of life?
So we don't know much about life expectancy at this point. So we do know that there are some Renew individuals that are alive in their 30s and 40s. We know of one that was diagnosed that died in their 40s of ovarian cancer. Whether or not that's linked is kind of impossible to tell. But individuals that are older are not going to have had genome sequencing. So it's actually very hard to tell at this point what might be the life expectancy, what we'd expect these individuals to die of and when. So it's definitely not early age life limiting like a lot of severe neurodevelopmental conditions are, but we don't yet know to what extent that's true.
And so there don't seem to be any other questions, so maybe I'll just keep having a chat with you about this.
That's great.
And so is there any compensation in expression of RNAu4-1 in any of these individuals, I guess? Because presumably, I mean, this is so important for, early development, and obviously there is, I mean, you can allow a certain amount of plasticity, but it seems like, you really need to kind of like, if this is the one that's expressed everywhere, you might expect to see, I guess, a more pleiotropic phenotype, is maybe what I'm asking, whereas you see mostly, I guess, a neurodevelopmental.
I always get asked this question.
At least for me, I mean, this is a classic question in the splicing field, right? Why the mutations in a, like, fundamental, we have the same with the helicase that unwinds these two, right? Why do mutations in that helicase lead mainly to retinitis pigmentosa?
Yeah.
So it's kind of like,
I have some theories. They might be rubbish, so we'll see. The theory around the RP predominant phenotype is that it's about slowed splicing rather than altered splicing.
Sure, yeah.
So the turnover of transcripts is important in the eye, and that's, I'm not an expert in that. Why is Renew predominantly a brain phenotype? Well, basically what we see here is systematic, global, but very low level disruption to splicing. So any individual splice site has a really small change. And that has to be true, because if it was a high level disruption, it would be embryonic lethal. It wouldn't be survivable.
Right, yeah.
So
yes, of course.
Where would that play out? Well, if you think about things like heart, the heart is like major structural proteins. If you varied them just a little bit, like, they probably wouldn't care. Where do we care about very careful, orchestrated co-expression of multiple different genes in the development of the brain? So I think that's why it's playing out predominantly as a brain phenotype, but there are also other multi-system involvement in the disorder. So yeah, but that's my theory.
Right. Yeah. Okay. We actually do have more questions that have come in now, so I'm gonna go through them one by one. So do the genes containing 5' splices that are differentially spliced consist mainly in neurodevelopmental associated genes? Which I guess follows on to the discussion we were just having.
Yeah, so I think this is complicated, right? So I don't think we should expect that we would be able to see that necessarily. And there are some, but every time you look at a gene set, there'll always be some that are neurodevelopmental. But I think it's actually more likely the ones that are changed the most are going to be the ones that can tolerate the change the most, which are unlikely to be the neurodevelopmental. So they're unlikely to be ones driving the phenotype, are the ones that we can actually observe in the data. So, and I think it's likely a disruption across multiple genes that's driving the phenotype, not one or two specific genes.
Right.
So,
and so I guess partly related to this are the variants in RNAu4 do they co-occur, or are they correlated with variants in other protein coding genes in these individuals, or is it just you know, there's not much of, I guess you would expect some because you have a disruption in the basic metabolism.
There's not any other genes that are recurrently disrupting these individuals as far as we can tell. Like, I mean, if there was a recurrent disruption in a protein coding gene, that would have been previously hypothesized as the cause of disease, rather than the RNU4 And so we've not systematically looked, but I think that would have been identified if it was true.
Right. And are there, I guess, actually related to this, are there limitations when you apply you know, these sequencing methodologies to the study of snRNA mutations? I'm not sure exactly what they just said when applying SGE to the study of snRNA mutations. I'm not quite sure exactly what they're asking here. But
we had to do the cut for the CRISPR outside of the gene because of the high homology across the different genes.
Right.
We'd risk mutating all of them, but as soon as you leave the gene body, the sequence is degenerate. So as long as you just declare with where you cut, then you can actually make the gene specific, the edit specific to RNU4.
Good. And I think we have one about therapies. So, I mean, I'll ask kind of a question before the question. So first of all, how do you see the therapeutics working here? Would you essentially be able to express, presumably you could express or inject very early during development kind of variant, an RNA that is already transcribed, and this might actually assemble. Is this kind of along the therapeutic angle, or what would be the?
So that's not the primary angle at the moment, but kind of what we think is the mechanism with the Renew syndrome is this mutant RNA is getting into the spliceosome and from there disrupting its function. We also think the mutant RNA is more stable than the wild type. We see a kind of 30% ratio in cells, well, in blood when we look at the RNA sequencing data, caveat. And I think that's because it's maybe more sticky in the spliceosome, and that's making it appear more stable because it's less available to be degraded. You might know that better, whether that's
that you could have stalled complexes that persist for longer. Although this is all very kind of tightly controlled, but that would be a possibility for sure.
So the kind of, because it's this kind of poison allele type mechanism, what we think about is kind of shifting the balance, right, between the mutant and wild type. What you could do is you could massively flood cells with wild type by expressing on some AAV vector and hope that wasn't toxic and it would work. But the approach that's majority being attempted at the moment is the opposite. So can we knock down selectively the mutant copy and leave the wild type?
Right, okay.
So there is multiple programs. So we're doing some work in Oxford, but there's also pharma companies. NLRM's a not-for-profit in the US that's working on this to design anti-oligonucleotides that selectively degrade the mutant RNA.
And so the actual question was whether this could become kind of a standard treatment for rare genetic disorders. I guess the question is, how many are they actually going to be, caused by snRNA mutations? But it seems like we're discovering quite a few.
And you need different. So the recessive is obviously loss of function. So for recessive, you need some kind of gene replacement. So it depends on the mechanisms, basically.
Let's see if we have any more that have come in. No, I think this is actually all of them. Which so actually, since you mentioned the recessive versus sort of dominant ones, it seems that mostly the ones that are it's the ones that are in these kind of regulatory regions, so the region that is important for this kind of keeping the Akagaga rightly positioned, it gets these mutations that are, sort of, you can get dominant mutations there, but like in the other cases, where you're affecting actual integrity, you'd always get sort of recessive, because those presumably would lead partially to some sort of embryonic lethality. And so that's actually, do we have this data, I guess maybe in the UK, from, I don't know, aborted fetuses, or that haven't been kind of, where you could have people, or have you considered looking also at the ones that are, kind of, that might tell us something about the ones that are really, really bad?
So most people that have done this, so definitely I think that there's some threshold of what's tolerated in terms of disruption, definitely. But most people that have looked at this or have these cohorts have done exome sequencing. So we can't look at the non-coding RNAs, which is slightly sad.
Like,
but there's an interesting thing that you might be, so for the recessive condition, we see exactly the same regions and actually sometimes exactly the same variants in RNU42 as we see in RNU4 attack, which also so they map almost perfectly.
Right.
But in RNU4 attack, you never get two mutations, so one in each allele in the SM protein binding site. The thought being that is complete loss of function and wouldn't be tolerated because you don't have another copy of RNU4 attack to compensate.
Yeah, I think that's
in RNU42, we do sometimes see a double hit in the SM protein binding site, because I think we have 4-1 that is able to compensate. So it's kind of very cool biology.
Yeah, well, and I guess maybe this could bring us to kind of a wrap-up thought that maybe part of the reason why we have so many duplication of these genes is that in the end, if you do incur mutations in these really important regions, you always you have a backup that you can sort of upregulate maybe, and that could be potentially a way, or maybe this is partly why we've kind of evolved to have so many variants. I think we can we're getting close to the end, and there are no more questions. Oh, right. No, there are no more questions. Sorry. Yeah. Sorry, there are no more questions. I thought one had come up just at the end, but so we can wrap it up. Thank you, Nicky, for a very interesting seminar, and for essentially opening up, I guess, a new avenue of understanding some of these neurodevelopmental disorders, and for bringing the spliceosome to the fore, which is dear to my heart. So thank you everyone for attending, and I think that's it for today.